Topic 7.4 Population Genetics and Topic 7.5 Hardy-Weinberg Equilibrium. So, evolution is driven by random occurrences, but Hardy-Weinberg's model describes and predicts allele frequencies in a non-evolving population. So, with Hardy-Weinberg, there are 5 assumptions, which are no natural selection, no mutations, no migrations, so that means like no emigration or immigration. We want large populations, and there must be random mating. Usually these conditions are rarely met, but they provide a valuable null hypothesis. So these are the 2 relevant equations for Hardy-Weinberg equilibriums. The first equation is used to find the genotypic or phenotypic frequencies of individuals in a population, where the second equation is used to determine the frequency of a particular allele in a population. So, p squared is the frequency of the homozygous dominant genotype, 2pq is the frequency of the heterozygous genotype, and then q squared is the frequency of the homozygous recessive genotype. So the conditions of Hardy-Weinberg when they're not met. So these are the factors that disrupt the equilibrium. So first we have mutations, which are changes in the genes that affect their expression. Non-random mating is when individuals choose to mate with one another based on a certain trait. Gene flow is when new genes are introduced to a population through immigration or emigration, and this usually leads to an increase of genetic variation. Genetic drift is the change in allele frequency within a population, which can occur due to random environmental events such as bottleneck or founder effect. And natural selection is when alleles improve or reduce fitness for individuals to survive and reproduce in a given environment. So, to expand on the idea of genetic drift, there are 2 types which we know of, which is bottleneck and founder's effect. So bottleneck is basically a reduction in a population due to a natural disaster, and that just leads to a few of the alleles to be lost. Whereas founder's effect is when a small group splits off from the main population and forms its own colony, which we can see with the pictures. And then some key points with genetic drift are that there are significant changes when it comes to a small population, because this can lead to an increased death rate and then low reproductive rates. And then there can be changes. This can cause it causes allele frequencies to change at random and causes a loss of genetic variation.